Hello, ladies and gentlemen. I welcome each and every one of us to this short introduction to Bioinformatics Part 1. My name is Dr. Ibuka David from the Department of Biochemistry and Molecular Biology, Faculty of Biological Science, AFUNAI. And this is coming to you from the Genomics and Bioinformatics group called DNA Analytica from the Department of Biochemistry. We are majorly on genes, genomes, bioinformatics. We work with DNA, with RNA, proteins. We do, you know, a lot of sequencing, mostly infectious diseases, and all that. All bioinformatics works and computational biochemistry. This is the group that handles this area. And here we'll be learning just the basics of bioinformatics. And to do that, central dogma is, you need to understand what central dogma is. And then we go to sequencing methods and certain sequence files. Then we look at gene sequence editing. How do you edit the gene sequence using bioedit software? And how do you build consensus sequence? Be able to learn how to build a consensus sequence and perform sequence alignment. And then we do blast and basic alignments. Then we look at evolutionary and phylogenetic trees. How do we compare our sequence with other existing closely related sequences and distantly related sequences? Okay. Um, you look at central dogma before. Central dogma has to do with, from DNA to RNA then to protein. You know, how do you do you know how much do you know about DNA, replication, transcription, and translation of DNA molecule? The information in DNA has to first be replicated, and you must understand what DNA molecules are, what RNA molecules are, and these are the major focus of the basic things you need to understand before you go directly into what bioinformatics are. And to understand that, you need to know what DNA molecules are made up of. The nucleic acids, first of all, every nucleic acids have certain nucleotide that it's built upon. Each nucleotide having three parts: the basis, the sugar molecule, and also the phosphate groups. And you know what the bases are: the purines, the pyrimidines, and you look at the ribose sugar and the deoxyribose, you look at the phosphate groups. And when this nucleotide is joined to another nucleotide, you form polynucleotides, which could be a DNA, could be an RNA. And these nucleotides are joined to each other by 3' 5' phosphodiester bonds. So you look at how this DNA are replicated from DNA polymerase into a major enzyme to another DNA, and how the DNA is transcribed to RNA, major enzyme and RNA polymerase. So how the information in the messenger RNA are used to you know to form the sequence of amino acids in a polypeptide chain of protein. So these are the central documents you need to understand and get to know this area properly. And also need to know that every gene has to be sequenced. In sequencing methods, we try to understand what the number of bases that are present in a gene and the arrangement of these bases. Of course, bases are the linguine, cytosine, thymine, uracil, In every gene, there is a number for the basis and there is the sequence in which it is arranged. So the sequencing method determines these two parameters in a particular gene. As when you have a DNA, you've extracted the microorganism, eukaryotes, and so on, you have to pass, it has to pass through a sequencing method. And one of the traditional sequencing methods is Sanger sequencing, also called dideoxynucleotide sequencing. You know, it has limitation because it can only sequence a short targeted gene, maybe gene for antimicrobial resistance, a gene for you know 16SR ribosomal DNA that are used to determine or differentiate different bacteria, ITS gene, differentiate between different fungi, short gene specific gene sequences. So it will be it will be laborious, time consuming to take weeks, months for you to use Sanger sequencing to sequence a whole genome. So in that case, Sanger sequence has a limitation. So in order to sequence a whole genome, maybe you want to look at the whole genome of a bacteria that is up to maybe 5,000, 5 mega base pairs, 5,000 base pairs, and you need to use a much more advanced techniques that's referred to as high throughput sequencing. These are also referred to as next generation sequencing or whole genome sequencing. It involves Illumina, nanopore, ion torrent, PacBio, solid. There are others. But one of the most common whole genome sequencing technique is Illumina. The Illumina MySeq, NextSeq, and so on. We want to look at bacterial genome, viral genome, bacteriophage genome. Illumina can give it to you in just a few hours. But when you want to look at more eukaryotic genome, large, Illumina can also do it, but its advice will go down to PacBio, nanopores, all that. They will give you longer reads. So having looked at all this, you know, you're able to look at these basic things and get to learn this. And in the next video, we'll dive into bioinformatics because with the knowledge of the sequencing techniques, knowledge of central dogma, then we'll dive into what bioinformatics are because we're looking at genes. So when we're looking at genes, you will not be, you know, confused. And that's I will I appreciate each and every one of us. Thank you for staying tuned and see you in the next parts of the video.